Hello and welcome to this next lesson in our topic of biological molecules for A-level biology. In previous lessons we've looked at carbohydrates, namely monosaccharides and their structure, disaccharides and polysaccharides. We're now going to start looking at lipids. So by the end of today's lesson you should be able to recall what a triglyceride is, you should know what a fatty acid is, you should be able to recall the structure of a phospholipid, and you should know how lipids are tested for. Okay, now as we would do for any other lesson, I'm going to write my notes on this screen here. I'm going to use the same colour coding that we always do, so we can start by defining what a lipid is. A lipid, L-I-P-I-D. A lipid, quite simply, can be defined as a fat or oil. Okay, so lipids are fats or oils, and lipids tend to have similar properties. So lipids have similar properties. So these properties include that they're insoluble in water. So they're insoluble in water. What that means is they don't dissolve. So they're insoluble in water. However, they are soluble in alcohols. Soluble in alcohols. And they can enhance the flavour of foods. So they enhance flavour of food. So for those of you that don't know what it means, you may wish to add this in your notes. I'm not going to add it on here. The word insoluble means that it won't dissolve. Soluble means that it does dissolve. So lipids don't dissolve in water, but they do dissolve in alcohols. Now, one thing that's really useful with lipids, and it is a property, but I'm going to add it in regs, it's important, is that lipids have large amounts of energy inside of them. In fact, lipids tend to have twice the amount of energy of a carbohydrate. So we can put that lipids, and that's the worst L I've ever seen, lipids have or provide over twice the amount of energy, of carbohydrates. And why this is important is that it means lots of energy can be stored in a small mass. So lots of energy is stored in a small space, And one way to think of this, I know we tend to think of biology as animals, but the best way to think of this is a seed. A seed of a plant is tiny, it has lots of oil in it, therefore it can store lots of energy in such a small space. So we can put here, e.g. seeds. Okay? Now, lipids tend to be made up, or are made up, of carbon, hydrogen and oxygen. However, what makes them different from carbohydrates is that they have far less oxygen in them compared to the amount of carbon and hydrogen in them. So we can say that lipids are made up of carbon, hydrogen, and oxygen. Atoms. But we can also say, but have less oxygen than carbohydrates. So lipids tend to have far less oxygen in them than carbohydrates. Okay? Right then, so let's have a quick think. Yeah, before we move on, we are going to define what an ester is. Ester, E-S-T-E-R. Now, an ester is a molecule with a carboxylic acid in it. So an ester is a molecule containing carboxylic acid. Carboxylic acid. And what we mean by that is that it's got this COOH in it. COOH. Okay? So I am going to refresh the screen. If you need any notes, get them down now, because we need to talk about the difference between triglycerides and phospholipids. So, if we refresh our screen, send it back to black, because it's easier to read that way, or I believe, and so I'm told. Let's start by defining triglycerides. Triglyceride. Now, a triglyceride can be defined as an ester, so it has this carboxylic acid in it, an ester formed from glycerol, and 3 fatty acids. 3 fatty acids. Now, this clue is given in the name. So, tri means 3. This is referring to the 3 fatty acids. And glyceride refers to this glycerol molecule. Okay? So we've got this glycerol and 3 fatty acids. Now, we could draw our glycerol molecule out, and our glycerol molecule consists of a hydrogen, 3 carbons in the middle. Remember, each carbon should have 4 bonds. A hydrogen should have 1, oxygen should have 2. So we've got our 3 carbons. We've got a hydrogen at the bottom. We've got a hydrogen up this side of the molecule. And on this side of the molecule, we've got our OH groups. Okay? We've got our OH groups. Now, this is glycerol. Okay, I'm going to write it at the top. I do appreciate there's not much room, but I'll do my best. This is our glycerol molecule. Our fatty acids, which I'm going to draw in orange, consist of a hydrogen bound to an oxygen, which in turn is bound to a carbon, which is double bound to an oxygen. we've then got this carbon being linked to other carbons, each of which have a hydrogen either side of it. Okay, I'm not going to draw it too long. However, these molecules would be massive in length. Okay, and we can fill in all the other sticks or all the other bonds with hydrogens. Okay, so we've got our hydrogens all the way around. Shake my hand off because it's starting to cramp, like so. Okay, this is our fatty acid tail. This is one fatty acid tail. In reality, a triglyceride consists of this glycerol molecule and 3 fatty acids. If you look, every carbon should have 4 bonds, every oxygen should have 2, and every hydrogen should have 1. So this carbon here, which is the one that causes a bit of confusion, has 1 bond here, 2 here, and 1 here, giving it 4. Okay? This would be our carboxylic acid group, and that's what gives this fatty acid its name. Okay, so this in orange is the fatty acid. So this around here, and I'm going to do it dashed, is our carboxylic acid. I'm going to have to put it down here. Carboxylic acid, and that's what makes this an ester. Now we can join this fatty acid tail. Remember, we would have 3 of them, so we would have 1 here, 1 here, and 1 here. We can join this fatty acid tail to our glycerol by removing water in a condensation reaction. So if we were going to remove water, we could remove H2O from here. So we're going to remove our water. I'm going to draw an arrow down here. We're going to remove our water molecule, H2O, and this central oxygen is then going to join the molecules together in what's known as an ester bond. Okay, an ester bond. So we would again have our glycerol, hydrogen, carbon, 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 and a hydrogen. I have to draw it. No, we need to be bigger than that, don't we? So we've got our hydrogen, our carbon, our carbon, our carbon, and our hydrogen. Can join this up. We've still got our glycerol molecule with its hydrogens on this side, and on the other side it's still got its OH groups, exactly like we drew up here, but this time we've removed water on this first carbon, so this is now going to join to the oxygen of the fatty acid tail, which is joined to a carbon, which is double bound to an oxygen, and then we've got our preceding carbons coming off. And through using a condensation reaction, we've removed a water and joined this fatty acid tail to the glycerol. Okay, we'll write all this down in a minute, but for now we'll just get our hydrogens drawn on, like so. Shake my hand off because this pen really makes my hand cramp. So we can say we've still got our carboxyl group. Yeah, we've still got this carboxyl group added to this glycerol molecule. So it is still an ester. Okay? So we can say, I'm going to write it over here, that fatty acid tails are added to the glycerol molecule by removing water, molecules in condensation reactions to form ester bonds. So on this diagram, on this molecule we've drawn, our ester bond, I'm trying to think which colour to use, our ester bond would be here. So we've removed the water molecule, joining the fatty acid to the glycerol to make an ester bond. Now this would happen 3 times to make a triglyceride, because it would have 3 fatty acid tails joined to the glycerol. So if we were to just, from now on, I'm not going to be drawing all this out because it takes forever. If we were to draw a triglyceride molecule, we would have our glycerol. So we've got our glycerol joined to the fatty acid tails via ester bonds. Okay, here's my fatty acids. So the orange boxes represent fatty acids. And our ester bonds, I'll draw, we used orange, didn't we? I'll draw in orange. So that's an ester bond. Okay, so we've said that a triglyceride is an ester formed from glycerol and 3 fatty acid tails. We've got a glycerol, which consists of 3 carbons, 3 oxygens, and then the 8 hydrogens. We've got a fatty acid, which has this carboxylic acid group with preceding hydrocarbons coming off it. We can remove water from the glycerol and the fatty acid to join them together to form an ester bond. A triglyceride would consist of 3 fatty acids joined to a glycerol by 3 ester bonds. So we've got a glycerol joined to 3 fatty acids by 3 ester bonds. Okay. I do need to clear the screen, so please pause the video if you still haven't got any of these notes down. You do need to be able to draw this, so make sure you learn the structure of a triglyceride or of a glycerol and a fatty acid. We're going to talk more about them in a moment, okay? So I need to refresh the screen. Please pause the video should you need to. That's the beauty of having a pre-recorded lesson. Let's refresh our screen back to black. And what we're going to do is, first of all, we're going to divide the screen into 2. About there, I'd say, is halfway. Very roughly. Oh, it's just between the K and the A on my MacBook. So, there are 2 types of triglyceride. First of all, we're going to talk about saturated fats. Saturated fats. Or, in fact, we'll put saturated triglycerides. Okay. A saturated triglyceride has no double bonds in its fatty acid tails. Okay, so no double bonds in its fatty acid tails, or in its fatty acids. So if there are no double bonds in the fatty acids, this is a saturated fat. Okay. So when we're talking about saturated fats or a saturated triglyceride, in its fatty acid tails, there are no double bonds. These tend to be found in animal fats. Okay? So these tend to be found in animal fats. Now, this means that the fatty acid tails are straight. So I'm going to put in red that saturated fats or saturated triglycerides results in straight fatty acids okay? This means that the triglycerides can pack close together and are solid at room temperature. So we can say, and I'm trying to think of a good colour to use, that the triglycerides can pack, sorry, you spelled can pack close together and are solid at room temperature. So saturated triglycerides or saturated fats, I'm going to add on there, you may wish to add on your own because I keep referring to them as fats. So saturated triglycerides or a saturated fat doesn't have double bonds in its fatty acids. These tend to be found in animal fats. It results in straight fatty acids and therefore they can pack close together, making them solid at room temperature. So in terms of their structure, you would have the glycerol, as we said earlier. So we've got our glycerol. Bound to this glycerol, we're going to have three fatty acid tails that are entirely straight. And I'll explain what I mean by that when we look at unsaturated. But they are entirely straight. They're joined with these ester bonds to the glycerol. So we've got our ester bonds. And we've got our fatty acid tails. Got our fatty acids. Now what that means is, if I was to draw another triglyceride down here, it could line up next to it and they would be of equal distance in between. They would stack up quite nice, okay? When we're talking about unsaturated fats, there's a double bond inside the fatty acid tail, and therefore it will kink upwards or downwards, and therefore you wouldn't be able to stack another triglyceride next to it. So let's have a think now about unsaturated. Unsaturated triglycerides or fats. Unsaturated triglycerides or unsaturated fats are fats with double bonds in their fatty acids. So they have double bonds in their fatty acids. These can either be mono unsaturated, which means one double bond, or poly unsaturated, which means more than one. Okay? So monosaturated, or monounsaturated, sorry. Monounsaturated means one double bond. Whereas polyunsaturated means more than one double bond. So polyunsaturated means more than one double bond. Just put DB, it means double bond, okay, just because I need the room. So when we're thinking about the fatty acid tail, we've got this OH group at the start, which is bound to our carbon, which is double bound to an oxygen, okay? One of these carbons would have a double bond in it, and when it has a double bond in it, it causes it to kink upwards or downwards. So we've got these carbons here, which have all got their hydrogens on. Then this carbon has got a double bond. This is a monounsaturated fatty acid. It's got one double bond in its structure, causing it to kink downwards. We are ignoring the carboxyl group, by the way, because this would be joined to a glycerol. We've got this downward kink due to this double bond. And I've done the schoolboy error of drawing a bond there. It's causing it to kink downwards. Okay. Now, when we're talking about room, and we're talking about triglycerides trying to stack together, this kink would prevent the next triglyceride or the next fat from being stacked next to it. Okay? So rather than having this straight molecule here, this one has now got a kink in it. So we've got our glycerol molecule, as we would do any other time, but instead of having straight fatty acids like we would have on a saturated triglyceride, we've now got fatty acids that kink upwards and downwards. They've got kinks in them. Okay, so these are my ester bonds again, joined to our glycerol, and we've got this kink here. So we can say that kinks in the fatty acids prevent molecules getting close from getting close, increasing the distance between them. Increasing the distance between them. Okay, so we prevent these molecules from getting close, and therefore they, due to the kinks, they can't get close, making a bigger distance between molecules if we stack them on top of each other. Olive oil is an example of an unsaturated fat. So olive oil is an example of this. So olive oil is unsaturated. Oh, what's happened? What's that all about? Go away. Oh. Let's just hope that this rectifies itself, because I really, really, really do not want to have to record all this again. So olive oil's an unsaturated fat. It has these kinks in its fatty acid tails which prevent the molecules from getting close, and therefore it is not a solid at room temperature but instead a liquid. It is an oil. Okay, let's see what's happening. I may have to force quit it. Oh, I think we're going to have to. So all I was going to write there is that olive oil is an unsaturated fat. We are going to have to force quit it. Just going to send the report. We don't want that to come up. We're going to reopen Sketchbook. Is it going to open? Here we go. And we can refresh it. Yeah? So, our next one we need to think about, or the final one we're going to talk about today. Apologies for that. I really didn't want it to stop recording, otherwise I'd have lost the will to live because I've had to record videos multiple times today. Our next one is a phospholipid. A phospholipid. Now, a phospholipid is defined as a lipid, or a lipid made of glycerol joined to two fatty acids and a phosphate group. And a phosphate group. Now, it's important to understand that phospholipids can be saturated and unsaturated. Okay? So when we spoke about saturation and unsaturation for triglycerides, phospholipids can be saturated or unsaturated. And that's really useful when we talk about their use inside of plasma membranes. Okay, so phospholipids make up phospholipid bilayers. Phospholipid bilayers. So phospholipids make up phospholipid bilayers. Now, phospholipids tend to have their glycerol, as they would do, as we've spoken about before. They will have ester bonds, which join their fatty acid tails to the glycerol. This time we're only going to draw two. You'll see why in a moment. So we've got our fatty acids, and we've got our ester bonds, except this time we're going to have an ester bond coming off the other side, joined to a phosphate group. Okay, joined to a phosphate group. Now, phospholipids make up phospholipid bilayers, and you often draw them like such. Rather than having the glycerol, you would have your phosphate groups drawn like this. So these are your phosphate groups. And coming off the phosphate groups, you would have your fatty acid tails. Okay? Except this time, we've got a saturated one here, and we can have an unsaturated one here. Okay, so these are our fatty acids, exactly like we've said here. This would be an example of a phospholipid with saturated fatty acids. So this is a saturated phospholipid. And this is a unsaturated phospholipid. Okay. Now, when we talk about the importance of saturated and unsaturated, that really shows when we're talking about phospholipids, which are used to make up phospholipid bilayers. If we were to draw a group of phospholipids side by side, so these are all saturated, they're all going to have straight fatty acid tails. If we were to then draw an unsaturated one, like so, because it's unsaturated, it's forced a gap between the phospholipids. Now, this gap is important because it creates fluidity, or it allows the membrane to be more fluid and more movement due to the gap between its phosphate heads. So we can just add down here, we can put a circle here, and we can say unsaturated phospholipids cause gaps or space in the membranes. So phospholipids are a lipid made up of glycerol joined to two fatty acids and a phosphate group. They can be saturated or unsaturated, and they make up phospholipid bilayers. Rather than having a glycerol joined to three fatty acid tails, you will now have a glycerol joined to two fatty acid tails and a phosphate group. These phosphate groups and the fatty acid tails make up a phospholipid. If they are saturated, the lipid tails are straight, whereas if they are unsaturated, you get a kink in them. These are important because they make space in the membranes. Okay? Final bit of this lesson. It's a bit extra, but it won't hurt you to know this. Cholesterol. Now, cholesterol can be defined as a lipid that is essential for health as it's vital in cell membranes. So cholesterol is a lipid essential for health as it is vital in cell membranes. Okay? Cholesterol is not soluble in water. Okay? So it's not soluble in water, and that's important to remember. So it's not soluble in water. Cholesterol can be added to a protein to make what's known as a lipoprotein. This makes them soluble. Okay? So we can define this as a soluble a soluble molecule which is a mixture of protein and cholesterol. Cholesterol is important inside of plasma membranes because it makes them fluid. Okay, I'm going to add in red. Cholesterol makes plasma membranes fluid. And you'll do more about that in your topic on plasma membranes. However, there is one thing I want to write down because it comes up quite a lot in exams and pupils tend to get stuck with it. So we can say that there are two types of lipoprotein. There are two types of lipoprotein. We've got what are known as low density lipoproteins. So low density lipoproteins. And essentially what these do is they are found inside of your bloodstream and they bind to receptors on cells. Once they bind to the receptors, they're absorbed and used in the membrane. But excess low-density lipoproteins are deposited in artery walls and form what are known as atheromas. So we can say, just to summarise that, we can say that they bind to receptors on cell membranes we can say that they then join the cell membrane but too many in the blood form atheromas on artery walls. But too many in the blood form atheromas, and I'll leave you to go away and look at what those are. Atheromas on artery walls. Therefore, we can say that LDLs or low density lipoproteins are bad. Okay? Whenever you come across LDLs and what we're going to talk about in a minute, HDLs, the LDLs are bad. On the other hand, high density lipoproteins, or HDLs are good. The reason they're good is because they have more protein than they do cholesterol. Therefore, they transport cholesterol from the body tissues to the liver where it's broken down, and it lowers blood cholesterol levels. So, HDLs transport blood cholesterol to the liver, to the liver to be broken down. So they transport blood cholesterol to the liver where it can be broken down. Therefore, it lowers blood cholesterol levels. So these lower blood cholesterol levels. So HDLs, or high density lipoproteins, are good. So HDLs are good. So just to summarise what we spoke about with cholesterols. Cholesterols are lipids that are essential for health and they're vital in cell membranes. The reason they're vital in cell membranes is because they keep plasma membranes fluid. They allow molecules to move inside of the plasma membrane. A lipoprotein is a soluble molecule which is a mixture of protein and cholesterol. There's two types of lipoprotein. Low density lipoproteins, or LDLs, which bind to receptors on cell membranes. They then join the cell membrane, but too many of them inside of the blood form atheromas on artery walls. These are bad, so LDLs are not good for your diet or not good for your body, whereas HDLs, or high density lipoproteins, transport blood cholesterol to the liver to be broken down. Therefore, they lower blood cholesterol levels and are good for you. So you should now be able to tell me what a triglyceride is, you should be able to tell me what a fatty acid is, you should be able to recall the structure of a phospholipid, and you're going to need to go away and look up how you can test for a lipid. Should you have any questions about this lesson or any other lessons, please contact me and I will get back to you as soon as I possibly can. Thank you for listening.